Okay, happy Wednesday afternoon. What we're gonna do now for today's afternoon session, I think, is have a look of more of a perspective on the types of assignments that I have assigned for us to complete this week. The earlier session that I offered today, the morning session, during that we reviewed the weekly material in quite a bit of a detail. So it's a little longer recording and it goes through some of the key points that you really need to know from this week's chapter. So I want to shift our focus for this recording to the homework and types of assignments and stuff that we have going on there. So we are now on week 3, as you can see from the learning module that I've opened there on the screen for you. Week 3 covers the chapters 2, 3, and 4 of our textbook. I provided some summary lecture videos of some of the key things such as basic chemistry of life, biochemistry, and cell biology as it relates to the chapter material. You'll be also able to find your PowerPoint files for the chapter material, and I provided a few different handouts. Most of these handouts I either refer in those pre-recorded lecture videos or during our revision session. So those are things that students have found useful. I'm always happy and open to find other resources that might help you, so don't hesitate to let me know if you have something on mind that would really help you with learning the chapter material. For example, the cell diagrams are a result of a student indicating that they found that those would have been useful for her. So I will deliver if you tell me what you need. That's something that I really very much enjoy as part of my job, helping you to excel in these classes. So on this week's materials, what I really have assigned for you has to do with the chapter quizzes on chapters 2 and 3. I do want to be fully transparent. Last week and this week are mainly kind of a review of some of the key concepts that you need to grasp and you need to have a good understanding of in order to succeed in your anatomy and physiology classes. So even though I have quizzes as homework for you from these chapters, we will not be testing you on the exam 1 on this material. So it's more of collect those points now, they add up towards your final grade. But once you feel comfortable with this material, I assume that we're ready to move onwards and we won't circle back to it anymore. So what I'm asking you to do this week as a homework are quiz for chapter 2 and chapter 3. Both of these are worth of 5 points and they're due at the end of this week. So what we're going to do, we're going to have a look at both of these quizzes, and I'm going to stop sharing just for a second so I can go to the right assignment. And again, you should be seeing a view of my screen. And Chapter 2 quiz really deals heavily with that basic chemistry, basic concepts that we need to grasp in order to excel on these subsequent anatomy and physiology classes that we're going to do. So let's have a look at some of the questions that could come up there. This first question that you're seeing is literally click and drag the labels to the right parts of the picture type of a question. So what we're seeing here, we're seeing multiple water molecules. And you remember with the water molecules, the electrons that were shared between oxygen and hydrogens were shared unevenly. These electrons spent more time around the oxygen atom. So as a result, there's a slightly negative charge at this end of the molecule, whereas the end where the hydrogen atom is located is slightly positively charged because the electrons spend a little shorter time at this end. So the overall charge of this part of the molecule is positive, whereas it was negative at the oxygen end. Because we had this slight difference how the charges are distributed in water molecule, that caused the negative portion of one water molecule being attracted to the positive portion of the next water molecule. And this bond that we see between water molecules was known as a hydrogen bond. Just to clarify, in terms of the bonds between oxygen and hydrogen in this molecule where we have two hydrogens, one oxygen, these bonds are going to be covalent bond. So this is the scenario that we saw on our chapter material where electrons are shared between oxygen and hydrogen in order to provide both of them a valence shell that's full. However, that's not a permanently full. There's that difference of time. So that's why we're seeing a polar characteristic of this molecule that where the electrons spend more time. So like we saw, water molecule consists of hydrogen and oxygen, two hydrogens and one oxygen. We saw that covalent bonds were all about sharing pairs of electrons on the valence shells of the atoms, such as in this case the oxygen and hydrogen, and the hydrogen bond was between water molecules that we're seeing here, for example. Let's have a look at the next question. Next question shows up as various different kinds of functional groups, and you're being asked, I believe, to drag these functional groups to where you would typically find them. So we'll see hydroxyl groups, typically found with sugars and alcohols, amino groups, characteristic for proteins, methyl groups, we often find those with fats, oils, and steroids. Phosphate groups are very characteristic for the nucleic acid. And then carboxyl groups, we typically find those with sugars and proteins. So that was that question. Moving forward on our quiz, we're seeing that we're being again asked to drop and drag and drop the terms to the correct categories. So here we're seeing some of the macromolecules that we discussed about. And for the carbohydrates, we see examples of carbohydrates could include, for example, glucose, sucrose, monosaccharides, and cellulose. For the proteins, what we're seeing that the amino acids are a good example of this, and we're seeing also the quaternary structure. You remember that we could study proteins at various different levels. So this is one example of that. For the lipids, the terms that fall in within there are fats and triglycerides. And your textbook discusses about these eicosanoids a little bit more. That's an interesting group and a worth of looking into. So that would fall under that heading. What we're seeing for the next question, we have 4 statements, and you're being asked to complete each of these statements with the correct term that completes it. So what we're seeing on the first statement, we're seeing that a large 20 metric ton rock sitting atop of hillside would be an example of potential energy. So you remember that was the type of energy that's stored in an object, but not actively released yet. To free the rock from the top of a perch, you need a catalyst, which could take the form of a person or a leveler to free the rock. So catalyst was whatever started the change that we were going to see that was going to take place. Once free the top of the perch, the rock demonstrates kinetic energy as it plummets through the valley floor. So kinetic energy was energy in motion. So as we see something moving, we're seeing it having kinetic energy. In the process of rolling down the hill, work was done to push aside trees and other stones that stood in the way. So you remember work was what was example of now that energy being put into use. Remember that energy can exist even though it's not implementing work at that very moment. The next question that we have here actually comes with an animation that you will watch, and then you'll be able to answer questions relating to that animation. So in this one, we're being asked which of the following binds to an active site of an enzyme, and that would be the substrate that bound to the active site of an enzyme. Remember, enzyme was something that assisted the chemical reaction or the reaction to happen, and in this case it was the substrate that bound to the active site of an enzyme. Good animation for you to watch. I definitely would encourage you. Here we have a matching question where you're being asked to match the elements of the body with their chemical symbol. So let's go through those. So the chemical symbol C stands for carbon, as we're seeing here. Chemical symbol H stands for hydrogen, as we're seeing here. Chemical symbol O stood for oxygen. Chemical symbol Cu stands for copper. And chemical symbol Co stands for cobalt. So again, a simple activity where you get to explore these one or two letter symbols that we use to describe different elements. Often they come from the English names of these elements, and there you have some examples, but in certain cases these abbreviations or symbols for the elements also come from Latin terms, so that's something that you'll see happening as well occasionally. The next one is tick box activity, where you will be choosing all of the true statements regarding DNA. So there's more than one correct statement. And the correct statements that we're seeing here that the DNA constitutes are genes. So gene was a section of a DNA that coded for a particular protein or for a particular trait. And that statement is going to be completely correct. We also see that the statement that DNA gives instructions for synthesizing all of the body's proteins is true. We discussed about that, how I used an analog that you would have a cookery book where you have many different recipes. That cookery book could be analogous to our DNA. So there's a lot of material in that cookery book. Not everything of it you will need, but some of it is useful, and some of it is useful at different times. So each recipe in this cookery book would be an analogous for a gene for a particular trait or for a particular protein. You never really cook every single recipe from a cookery book all at once. You choose the recipes that are most useful at that time. And that's exactly what we do with our DNA. We activate genes that we need at that time. And when you take a recipe, so a gene, that gives us instructions how to make a dish. And the dish would be analogous to what we're seeing as a protein in case of this analog that we're using. So I hope that that helped to kind of make a little bit of sense of that. The next question asks us that RNA plays role in which of the following, and the correct answer is that RNA is important in synthesizing proteins and copying instructions from DNA. So as you remember, DNA was the string of instructions that always stayed within the nucleus of an eukaryotic cell. DNA never just wanders off from the nucleus. However, that DNA can be copied. We can make a copy of a section of the DNA that's useful for us at that time. And that copy would be considered as our RNA. So we're copying a section of that, and that RNA can actually leave the nucleus and then travel into cytoplasm to find ribosome that then helps us to synthesize the amino acid chain based on the instructions of that RNA, and that amino acid chain becomes a protein then for us. I think an analog that I used during the lecture was to think of a DNA almost as a very valuable manuscript that we might have in a special collections department of a library. You're not going to be able to check out that manuscript, but you can definitely come to the library to read that manuscript and study it. You can take notes of it. You can even take a copy of it. And that copy is now our RNA. RNA you can take with you, so the copy you can take with you out of the library or special collection, so you can take it to the other parts of the cell, and based on those instructions, now we could do them proteins on those instructions that we copied from the DNA and carried out as an RNA. The next question is again a multiple choice question. Which of the following statements is correct regarding the biological properties of water? We will see that not only does water ionize many other chemicals such as acids and salts, but water itself ionizes into hydrogen and OH. So we're seeing that as well as an example there. There's a true-false statement. Catabolism consists of energy-releasing decomposition reactions. I made a little sketch during our review session to explain the concept of catabolism and anabolism. So catabolism does indeed consist of energy-releasing decomposition reactions. That is true. So we're seeing when we take a complex molecule and breaking it into smaller parts, as we're breaking those bonds that held the complex molecule together, we're releasing energy. The next question, again, I think that provides you a little animation that you can watch, and then a follow-up question. And the follow-up question that we're seeing is what is the name of the positively charged subatomic particles? And we saw that protons were positively charged subatomic particles, whereas neutrons were neutral, there was no charge with them, and electrons were negatively charged. So that was kind of a nice question there. There's a second question that we see here, which asks you, is it true that the number of electrons in the valence shell determines what types of bonds an atom can form? And that is absolutely true. You remember valence shell was the outermost shell of an atom. So the number of electrons there defines really whether that atom is reactive or stable. If it was reactive, that simply meant that that atom was close of getting a full valence shell, so it was very eager to take part on chemical reactions in order to accomplish this. If an atom was very stable, it meant that it typically had a full valence shell, so the maximum number of electrons then can fit into that outermost shell, and as a result, it was not very eager to take part on chemical reactions. So I think that that should help with that question. I hope so. Let's have a look at the next question. Here we are being asked to match elements with their chemical symbols. So we see symbol C for carbon, Mg standing for magnesium, Cl standing for chlorine, Na for sodium, Fe for iron, and K for potassium. And again, you will become more familiar with this. More of those you work through, but the periodic table of elements often relies on these chemical symbols. Then we have a question where we're seeing in the following reaction, what is or are the products? So we're seeing this material going in, and then we're seeing an arrow which tells us that these are the reactants that are on the left-hand side of this reaction. And what's on the right-hand side are going to be the products, the outcome of this reaction. So we have our reactants, what is going in, what's needed to start this reaction on the left-hand side, and on the right-hand side, we will see the products, what comes out of that reaction. So I hope that that helps with that. Here we have various different properties of water, and we're matching them with their function. So we're seeing, for example, that solvency describes how molecules must be dissolved in water to move freely and react. Cohesion told us about how water molecules have a tendency to bind tightly, cling tightly to other water molecules. So cohesion really was the attraction of water molecules to other water molecules, which then allowed us to have things like surface tension and so on. Next one that we're seeing is adhesion. Adhesion tells us how water lubricates organ surfaces and their surrounding pleura. Adhesion really described how water molecules tend to be attracted to other polar surfaces. So, for example, the humidity on the surfaces of plants or spider webs in the morning would be a good example of that, as is this statement that we're seeing here. The next one that we're seeing, we're being told that water ionizes and can be incorporated into other molecules, which really tells us about the chemical reactivity of the water. Water is very much able and eager to take part on different chemical reactions. And finally, we're seeing the thermal stability as the high heat capacity that helps maintain the internal body temperature. So we saw that water was excellent in binding heat and then releasing it slowly. This is why we don't see huge temperature fluctuations if we live by the beach. Usually during the hot day, the big bodies of water tend to capture some of that heat, and during the colder nights, they tend to release slowly that heat. So their temperature fluctuations are lesser than, for example, what we can see here in Arizona, where it's really dry and we go through these extremes, really hot days and really cold nights quite easily. We also saw this thermal binding ability of water in how we were able to transport heat from one body part to another body part. That was a good example of that as well. The final question that I have for this quiz asks that the energy that is stored in the bonds of a glucose molecule exemplifies of which of the following, and there's multiple correct answers. So these bonds within the larger glucose molecule really hold chemical energy. So chemical energy in large molecules is stored in a way how these individual atoms come together to form the larger molecules in these bonds. And these bonds also, in that sense, if you think about it, are an example of potential energy. Energy that's a present there, but not being used at this very moment. So that completes our quiz for the chapter 2. And we will have a look of quiz for chapter 3 as well. I think it's going to be a useful activity for us to have an idea of what kind of questions come up on that. So let me share my screen once more. And on here, we'll see that we've got 11 questions. Again, the first question shows us 4 statements, and you're challenged with choosing a correct term to complete, or correct terms to complete those statements. So the first statement reads that cells are typically measured in micrometers, which is 1/1,000,000th of a meter. So we saw how cells were generally very, very small, and that small size of the cells was dictated by the surface area versus volume ratio, which really showed us, as we discussed, that cells need to remain small in size. We're also seeing a statement where we're reading that cells can only grow to a certain size in part due to the relationship between its volume and its surface area. So that's the concept that I was just referring in terms of the surface area versus volume ratio. We see as the cell size grows, the volume of the cell grows at a faster rate than the surface area of the cell. And we really want to have a lot of surface area for a cell so that there's a chance to do a lot of exchange of materials across the cell surface. Here we have a statement that says an egg cell is one of the few human cells that can be seen with the naked eye. So even though most cells are so small that you need a microscope or other magnifying tools to see them, there are few that we can see with a naked eye, and an egg cell is definitely one example of that. Finally, we're seeing a sentence where it says, since the volume of a cell increases faster than the surface area, if a cell grows too large, it runs the risk of rupturing. And that is very much true, and again binds into the surface area versus volume ratio that we discussed in the couple of earlier statements that we saw for this question. So I hope that that was kind of making more sense of that kind of a question. Here we have a question, which membrane-bound organelle is the site of protein and lipid synthesis? And we see that endoplasmic reticulum was where we had a lot of the protein and lipid synthesis happening. So this is where steroids and other lipids are synthesized, detoxification of alcohol and other drugs, manufacturing nearly all membranes of cell, and rough ER contains those ribosomes on the surface, whereas smooth ER did not, and it was nice and smooth. Here we have various pictures, and you're being asked to click and drag each label to identify the organs that we're seeing. First of all, we're here seeing the nucleus, which contained DNA. Then we're seeing smooth endoplasmic reticulum, which, for example, was involved with detoxifying alcohol in liver cells. We're also seeing rough endoplasmic reticulum, which was characterized by having lots of ribosomes on its surface. Then we're seeing a Golgi apparatus or Golgi complex. This enclosed proteins in vesicles. So remember, it worked a little bit like a distribution center of a trucking company. So materials come in, they get sorted, they get repackaged, and then sent away. That's what Golgi apparatus or Golgi complex does. We're also seeing lysosome here. There's a small sac-like structure that contains digestive enzymes in this example. Here we're seeing a picture of mitochondrion, often described as the powerhouse of the cell. So that's where the ATP was produced from the sugars and oxygen, and that ATP was the cellular currency of the cellular energy currency that the cells used. Final picture, what we're seeing here is a centriole, which played an important role during the cell division in mitosis, for example. So we're seeing all kinds of cell organelles here. Remember, cell contained various different kinds, and this week is really a good chance for us to remind ourselves of those. We're seeing some of the more detailed questions here. In terms of that, let's start with smooth endoplasmic reticulum. This organelle was really important, for example, for synthesis of phospholipids. We're also seeing rough endoplasmic reticulum, and this portion of the ER was where protein synthesis took place. And we're seeing the ribosomes forming the kind of rough surface of the rough ER. And ribosomes were important because this is where the protein synthesis took place based on the instructions that we got from the RNA. We can also see some cisterns here, these folded, flattened sacs. They are present on both smooth and rough ER. And again, I think that that's a nice review of some of the structures that we need to see here. Here we're seeing a diagram or a drawing of a cell, and we're being asked for the highlighted organelle in this picture, highlighted on green color. And what we're seeing here, we are seeing our Golgi apparatus, which were really important for regulating the transport and modifying newly synthesized proteins. So again, think of that almost like a distribution center of a trucking company. So an interesting structure in that sense. Another structure that's highlighted on the following picture, here we're seeing the mitochondria highlighted. And we remember that mitochondria were the powerhouse of the cell, if you wish. So this is where ATP is being produced as part of the process of cellular respiration. So we needed sugars, we needed oxygen, we produced ATP and carbon dioxide and some waste products as well. What we're seeing here on this image, on this highlighted structure, we're really seeing the endoplasmic reticulum. And when asked about the functions, the correct option that we're seeing here is that this is where we're synthesizing protein for secretion, insertion into plasma membrane, and lysosomal enzymes. So a good structure in that sense as well for us to see. Here we have a sentence that has a gap, and we're asking you to complete this gap by typing in the correct answer to the box. So in this question, we're being asked that the organelles that read coded genetic message from the RNA and assemble amino acids into order based on that genetic message, and these amino acids then became proteins. These organelles that were responsible for this were our ribosomes. So they're these small structures that converted the RNA into amino acid chain, and amino acid chain then became a protein. Here we're seeing a statement, and we have a multiple choice question. Mitochondria extract energy from which organic compounds and transfer it to which of the mitochondria extract energy from organic compounds and transfer it to which of the following for the use of by the cell, and we saw that mitochondria produced ATP. So they are the cell organelle responsible for synthesizing ATP. On the next question, again, we have a nice little animation first, and then you get to answer a question. Here we're seeing a question about exocytosis. And exocytosis is a process by which cell releases substances from the cell via vesicles. The way I think about it is that since it's exocytosis, it's like exiting the cell. So that might be one way to think about it. There's a follow-up question. Certain white blood cells engulf microorganisms and bring them into the digest them. This process is best described as phagocytosis. So in phagocytosis, cell comes and engulfs something that should be removed from the body. So these phagocytotic cells are really cell-eating cells. They engulf something that shouldn't be around, and then as they engulf it, they can then break it apart and eliminate it from the body. I think that was the last question on this particular question, and that brings us to the end of that quiz. I think it's been a useful activity to have a look of some of these questions and have an idea of what you should complete for this week. Remember that I always offer also additional assignments for any student who wants to go above and beyond. These are not required for this course, but if for some reason you feel like tackling them, please don't hesitate to submit them as well. I'll be more than glad to give you extra credit for completion of those. So that wraps up everything I had planned for this session. Like I said, I have a longer recording from this morning where I review in great detail some of the key points of this week's chapters. And next week, we'll actually get to the actual material. We'll start looking at the first system of the body. And the first system that we will be looking on week 4 is going to be the integumentary system. So the skin, glands, hair, nails, other associated structures that go with that. But at this point, I think that this is a good place for us to stop. I hope you have a very good week. Remember that I'm here available for you in many different ways to assist you. All you have to do is reach out to me, and I'll be more than glad to find a time and place and method that works for both of us. As always, I do have my office hours. I do have these live online sessions. I appreciate you being part of this class, and I really hope you have a good week, and I look forward to continuing to learn with you. Have a good rest of the week. Thank you and bye.